Hi, everybody. So now we're going to talk about organic molecules and how they differ from inorganic molecules. Organic molecules are your molecules that contain carbon and hydrogen. Inorganic molecules do not contain both carbon and hydrogen. In general, they don't even contain carbon, but there are a couple of examples of carbon-containing inorganic molecules, which is why I say that all organic molecules always contain carbon and hydrogen. So the four important organic molecules that make up living things are carbohydrates, fats, proteins, and nucleic acids. Macromolecule is just a super large organic molecule. A macromolecule is also known as a polymer. A polymer is just a large macromolecule that is made up of small subunits that typically are either going to be identical or similar. So a monomer is a small molecule that builds up larger polymers. The term polymerization is just joining monomers to form the macromolecules. And we do this through dehydration synthesis, which I've already talked about. Dehydration synthesis is the formation of a bond while taking out water. So dehydration, removal of water, synthesis, forming a bond and producing something. Hydrolysis is how we break apart macromolecules. And so we do this by adding water back in. So hydro, water, lysis, break down. And so we're going to break bonds apart through hydrolysis. And we add water back into those molecules. Here's an example of dehydration synthesis. One monomer and another monomer come together. They form what we call a dimer, or a small bonded molecule. And we produce water as a subunit. So dehydration synthesis produces a molecule. Hydrolysis breaks it apart. So if we're going to break the dimer apart, we add water back in and we produce each of the individual monomers. So let's talk about those four macromolecules. The first one is carbohydrates. Carbohydrate gives us the energy that we use for everyday work. The energy that we take in, carbohydrates, typically is converted to glucose, and glucose is then digested and converted to, or I won't say that, and oxidized to produce ATP through cell respiration. There are three types of monosaccharides that we typically talk about: glucose, which makes up our blood sugar; fructose, which is fruit sugar, and it's sweet; and galactose. Disaccharides are produced when two monosaccharides come together through dehydration synthesis, and they form that dimer. Sucrose is table sugar, and it's formed of glucose and fructose. Lactose is milk sugar formed from glucose and galactose. And maltose is grain sugar formed from glucose and glucose. Then we have polysaccharides. These are the large polymers that make up those large carbohydrates. The three that we typically focus on are glycogen, which is animal storage of carbohydrates. So think about when you ate 3 jelly-filled donuts. That's a lot of sugar entering your blood. We don't need that much sugar. And so most of that sugar is going to be taken out of our blood and stored in our liver and in our muscles as glycogen. Starch is plant storage of their glucose. So they're going to take the glucose, form starch molecules that they can use later on when they need energy. And we use starch as a food source to gain more glucose. And then cellulose is plant form of or a plant form that forms the structure of the cell walls, and we cannot digest that form of carbohydrate. So we use cellulose as a fiber that helps to push out other waste products through our intestines and out of our system. So it's a fiber. I just talked about this, but glycogen, energy storage for animals, starch, energy storage for plants, and cellulose, structural molecule that forms plant cell walls. We also have some conjugated carbohydrates. A conjugated carbohydrate is a carbohydrate that has a protein or a lipid associated with it. So glycolipids, we have a carbohydrate bonded to the lipid. Or a glycoprotein, we have a carbohydrate bonded to a protein. Proteoglycans are carbohydrates and proteins that form a jelly-like substance that helps to hold cells and tissues together. Now we'll move on to lipids. Lipids are hydrophobic organic molecules. They are composed of carbon, hydrogen, and oxygen, but they have a very high hydrogen to oxygen ratio. Whereas in carbohydrates, it was a 1 to 2 to 1 ratio, carbon, hydrogen, oxygen. In this case, you have a lot more hydrogens compared to the oxygens that are there. Lipids are not oxidized as easily or as readily as carbohydrates. So because of this, you can get a lot more energy per lipid. The 5 types of lipids or fats that we have are fatty acids, triglycerides, phospholipids, eicosanoids, and steroids. So this is what a fatty acid looks like. A fatty acid contains a polar functional group over here, and then it has a nonpolar tail. There's polar functional group and nonpolar tail. You see this one has hydrogens surrounding all the carbons, whereas this one has hydrogen surrounding most of the carbons, but not everywhere. So there's a double bond here. Fatty acids are called amphipathic molecules because they have a polar region here and a nonpolar region here. Amphipathic molecules have 2 different charged groups, or 2 different types of groups. So you have the charged on one side, polar on one side, and nonpolar on the other. And so they're going to interact differently with water depending on which part we're talking about. And that's what I just said. If fatty acids have hydrogens surrounding all of the carbons on all sides, then they are called saturated fats. If there's a double bond anywhere in the fatty acid chain, then it's called an unsaturated fat. So saturated fats are typically seen in animals, in animal fats, whereas unsaturated fats are typically seen in plants. Triglycerides are fats found in animals and plants, and they're formed through dehydration synthesis. When they are liquid at room temperature, they are plant fats, plant triglycerides. And if they are solid at room temperature, then they are animal fats. And so liquids tend to be unsaturated, plants tend to be saturated. The function of fat is for energy storage for long term. So we're not talking about like the glycogen granules that we store in our liver that will allow us to break down, or will be broken down rapidly when we need more sugar in our diet, in our blood. These are for long-term storage. So you can store this fat for decades. It functions in insulation, so the fat warms the body. It acts as a shock absorber, so it surrounds organs, and then when those organs, any damage to that region of the body, the fat will take in as much of the shock from the damage first, and that protects the organs that are deeper. So we form triglycerides. Our triglyceride is made up of a glycerol and 3 fatty acid tails, and they're linked through dehydration synthesis. They can be broken down through hydrolysis. So now let's look at phospholipids. Phospholipids are some of the most important fats in our body, and the reason I say this is because they make up every one of our cell plasma membranes. So without phospholipids, we would not have the plasma membrane of our cell, and if we don't have the plasma membrane of our cell, then we don't have cells. They're basically the outer, the walls, if you want to put it as, of the cell. They make up the structure of the cell. Phospholipids are amphipathic, just like triglycerides. They have a glycerol, which is polar, and then they have the fatty acid tails, which are nonpolar. Eicosanoids are fats that act like hormones, but they're not produced by endocrine glands or endocrine tissue, and they don't tend to go into the blood and travel to the neighbor or travel to cells that are far away. They tend to leave one cell and they enter another cell that is right there, a neighboring cell. So they help in producing inflammatory responses or help in telling neighboring cells to switch on certain characteristics, to turn on certain characteristics and turn off other characteristics. Steroids are fats that act as hormones. So steroids are all produced by one parent steroid, which is called cholesterol. And steroid molecules move into the blood. They bind to protein carriers, and then they move around the body and they interact with specific cells to tell those cells to either turn on or off different characteristics. Cortisol is an example of a steroid that acts on our cells and tells our cells not to utilize glucose. Estrogen and testosterone are hormones that enter our blood, and they activate secondary sexual characteristics as well as activating spermatogenesis and oogenesis. The majority of the steroid molecules that we have, and the cholesterol that we have, is synthesized by our liver already. We need very little cholesterol or fat added to our diet. The majority comes directly from our body synthesizing it. So this is a table that shows different steroid activity or different fat, I won't say steroid, fat activity, so the functions and the different types of fats that we have here. Okay. Nucleic acids then make up the basically the building blocks of who we are. So nucleic acids have a nitrogenous base made up of either adenine, guanine, cytosine, thymine, or if we are RNA nucleic acid, then we have adenine, guanine, cytosine, and uracil, not thymine. We have a sugar, depending on the type, It's going to be either deoxyribose for DNA or ribose for RNA. And then we have one or more phosphates. One nucleotide that makes up, or that is made up of an adenine and a ribose with a 3 phosphate groups is called adenosine triphosphate, or ATP. ATP is what gives our cells energy. So it's the energy currency for all of our body cells and other organism cells. So like bacteria use ATP, plants use ATP, we all use ATP. ATP is energy, okay, that our cells can use to build up other things or break down things depending on what we're doing. ATP can be broken down. So we break down the bond between the second and third phosphate, and we release the energy, and that energy can be used to drive other reactions. We can add the phosphate back to the ADP, adenosine diphosphate, with 2 phosphates, producing more ATP. Another molecule that I always talk about is cyclic AMP. So with cyclic AMP, we have adenine again and ribose, but we only have 1 phosphate, and we form a cyclic molecule. Cyclic AMP, also known as CAMP, is important because it acts as a second messenger in the endocrine system for protein hormones. I always talk about it in chapter 2, and then I always bring it back up in chapter 17 in second semester. And most people don't remember it, but I'll do it again here. I'm mentioning it, and we'll see if you can remember cyclic AMP as our second messenger in the protein mechanism for water-soluble hormones, okay? So the larger molecules are DNA and RNA. DNA, deoxyribonucleic acid, is a polymer made up of multiple monomers, adenine, thymine, guanine, and cytosine. And DNA is found in our cells, in the nucleus of our cells. It basically carries the information, the instructions for building every single protein that our body has. So it's the hereditary information that makes up our proteins in our body. But if DNA makes up our proteins, then RNA is the workhorse, actually makes the proteins. So DNA carries the information. It's like the architect that builds that pretty picture and says, this is what we're going to do. And then he has all of the workers actually do the work, and he sits back in his office. DNA is like that, the architect, and the actual workers, the builders, are the RNA molecules. RNA then is single-stranded, DNA is double-stranded. RNA can be found in 3 different forms as messenger RNA, ribosomal RNA, or transfer RNA. It contains ribose instead of deoxyribose, which is found in DNA. And RNA contains uracil instead of thymine. RNA is what makes up, or what actually builds the proteins, while DNA carries the information for building the proteins. I'm going to stop on this video here because we're at 19 minutes and I don't want to overwhelm you in one video. So we'll stop here and then we'll pick up with proteins in our next video. Bye.